So, the X-Men by Stan Lee and Jack Kirby for the X-Men #1 in 1963. And the original X-Men were just a group of 5 teenagers, and that comic never really sold very well. So eventually, it was canceled and blah, blah, blah. Eventually, they were reinvented in Giant Size X-Men #1 in 1975 by Len Wein and Dave Cockrum, and this gave us the cast of characters that are so dramatically ridiculous. So the originals were just a bunch of kids in matching onesies. But then eventually it was reinvented to be a bunch of international characters with somewhat more dramatic powers and more diverse worldview. The whole point of X-Men is what it means to be different, right? So they really nailed the idea there by reinventing with international people. So mutants from all over the world with extremely varied powers. The fact that they're from all over the world tells us a lot about whatever is causing the mutations is all over the world, so that's interesting right away. The fact that they have extremely varied powers, that becomes difficult to explain, right? One thing causing all of these different things.
And one gene in particular.
And one gene, the X gene. So anyway, what is a mutation then? Okay, we already sort of talked about it. Let's look a little deeper at the issue of mutation. First off, what is DNA and how does it work? All right, this is important for evolution. It's also important for next class when we talk about augmenting someone. We'll talk about CRISPR, genetic engineering, so we need to know how DNA works. All right, so you probably know this sort of thing. This is DNA. Hello, DNA. Proven by Watson and Crick in 1953, although there was a lot of work leading up to that. It is a double helix made of repeating nucleotides. The nucleotide chunks, there's 2 nucleotides on each side, and then you repeat it to make each stair of the staircase. All right, so the nucleotides are deoxyribonucleic acid. All right, deoxyribonucleic acid, DNA. And there's 4 possible versions of the DNA nucleotides, 4 possible bases on them. So here's the nucleotide chunk, okay? So you got one here, that makes a stair. Then you got another and another that makes the next stair, and another and another and another and so forth. So it is a chain. So in the nucleotide, this chunk, the chunk of the stair, so you can see the nucleotide is a bit of the backbone, right? And then also this chunk of the stair, half of the stair, that's called the base. And there are 4 possible bases: thymine, adenine, cytosine, and guanine. All right, TACG. All right, making up the 4 letters of the DNA genetic code, Gattaca. All right, TACG, ACGT, whatever. All right. So the fact that DNA has this, so you can read it off, right? A, T, C, G, C, A, and so forth. So you can read it off. It can contain information, just like you can read off information off a page, or you can read off binary from a computer. 1s and 0s in a computer, 26 letters in the English alphabet, 4 in DNA. So it's useful for containing information. All right. So here is if you unwind the helical staircase, right, to just make a ladder, then you can see side by side the two chains. A bond, you can see A has a shape to it that fits into T, and vice versa. C has a shape that only matches with G. They cannot connect otherwise. So A bonds only with T, G bonds only with C, and vice versa. And that's the rule of genetic pairing. So if I gave you the left half, you could make the right half, right? So let's say the left half reads ACGT. The right half will therefore read what? And so TGCA, ACGT, TACG, or GTAC. Which, if the left half is ACGT, what will the right half be?
No.
Okay. Got some variety of the votes. Cards up, cards up.
No.
Okay.
I don't know.
Well, okay, so let's go through it, okay? A, so bonds only with T. So it's got to be A or C right away, right? Most of you are going C, so let's see. A bonds only with T, so T. C bonds only with G, so TG, and we're done. I don't even need to really care about the other letters, but again, G bonds only with C, T bonds only with A. Okay, so most of you, I guess, were wrong. TGCA, all right? The beauty of this, you don't even need to do the full question, right? 2 or 3 letters will give you the answer in a multiple choice format. And you better believe you'll see a multiple choice format. Okay, so there you go. You can take the left half, you can make the right half. If you only had half, you can remake the full. What that means is you can rip it in half and make 2. You can copy it. And that's the other very, very important part of DNA. It can contain information and be copied. All right, that's huge. Along with DNA in our cells, we also use RNA, ribonucleic acid. All right, ribose nucleic acid is similar. Okay, obviously one difference is deoxyribose. Instead of deoxyribose, you got ribose. That's why it's RNA. It's also single-stranded, as you can see. It does not come double-stranded. It's only single-stranded. And one other big difference is the letters of RNA are C, G, A, and U. So anywhere you would have a thymine in RNA, you instead have a uracil. Okay? Anywhere there would be a T, you instead have a uracil. You just swap it out. So RNA is somewhat simpler. It's not as good. All right? It's not as it's more prone to errors when you copy it. And it's a little loose, as you can see all these dangly bits, right? So it's a little less stable. As a result, what we actually think is that life on Earth arose using RNA, and then eventually a couple changes happened, and we unlocked something even better, DNA arose. And as soon as that happened, we had life on Earth full. So there was actually a time, we think, where a time called RNA world, where the world was full of living things using RNA. But then once DNA was invented, whatever single-celled critter did that, took over the world because it had a much better molecule. But it still used RNA, right? It still had RNA inside of it, and so we all still do. Yeah, Eric?
Yeah, I was just going to ask why RNA is more difficult to copy.
I don't think it's more difficult to copy necessarily,
Okay, so
but it's not part of the copy process for us. When we need to copy things, we use DNA because it is less prone to errors because it's double-sided.
Okay.
Right? You have to screw up both sides pretty much. So anyway, that's the use of DNA, is that it can be copied. You can split it in half and then create 2, right? Because you can autocomplete. Using the left, you can autocomplete the right. Using the right, you can autocomplete the left. All right? This is more accurate than copying data on a computer. So if you think about all the times you've moved a file over to a USB drive, have you ever done that and then the file no longer worked? Because honestly, I never have. There's issues sometimes, but I've never transferred a file and it had a typo in it now.
Yeah.
You know, that doesn't happen. But it can. DNA copying is more reliable than that. Okay? So DNA copying is super, super effective. Evolution has worked very hard to make sure it is. All right? Because otherwise life would not function.
It's almost like it's the building block of...
It's almost like it's important to us, right? So yeah, it contains information coded basically like binary on a computer, or the letters in English, but only 4 letters, right? How is it used? How is it, I'm sure you know what DNA is, broadly speaking, and we've talked about evolution. How is DNA actually used? A, it determines what you look like on the outside. B, it's blueprints for building your cell structures. Or C, it's a recipe for building your proteins. What do you think?
Oh, this is a thing. Wait, what?
So most people have no idea.
I don't know.
So most of you are voting B here. C is the answer, all right? Which got a couple votes for C. So recipe for building your proteins. That's it. It does one thing. It tells you how to build proteins. That's it. It does not at all say what you look on the outside, all right? In fact, if I took your DNA, there is no way in hell I would be able to figure out what you look like from that, okay? Which is why, for example, cops cannot do that. They cannot take crime scene DNA and then figure out what the suspect looked like or whatever. Because it tells them what the proteins inside the suspect look like. Okay, how's that?
It doesn't give you an exact copy.
Well, so it is the code for building proteins, all right? Proteins are the tools, they are the functional components of a cell. They are the things that do stuff in your cells. The tools and building blocks of all cells. There is no time where it's more important to have the right tools than when you are building something from the ground up. So when you start off with a fertilized egg and it starts to divide, the proteins that are there will determine how it's built, what shape it acquires, what features it has, right? So the tools determine the person, but it is a long route to get there, all right? So how does that actually happen? Now, this is pretty complicated, but I want to walk through it because it's one thing to say it builds your proteins, but I want to actually show you how it happens. This is complicated, so I will ask you a question on the test about this, all right? So look it over again. It's a process called DNA transcription and translation. The first step is we are going to transcribe a piece of RNA, which will take the information from the DNA over to a factory. So this is called the messenger RNA, the messenger or mRNA strand. So we unzip our DNA, okay? We open it up and we read one side of it. We transcribe a message from that into RNA. So if there's a T, I'm going to use an A in the RNA. If there's a G, I'm going to use a C. If there's an A, I'm going to use a T. But this is RNA, so I'm going to use uracil, U. T goes to A, C goes to G, G goes to C, and so forth and so forth. All right? So you end up transcribing a message based on what was in the DNA. All right? The mRNA is taken over to a chemical factory called the ribosome, which is a true nanotechnology that we find in our cells, a natural nanotech factory. The ribosome will do the hard work. So here's the ribosome, it's very complicated. So the way this is going to work is the strand of messenger RNA, or mRNA, will be fed into the ribosome. Every 3 letters are going to be looked at, right? Every 3 letters, AGU, CGU, ACG, UCA. It comes in 3s. I don't know why, but it does. All right? So we're going to feed it in, and every 3 letters we're going to look at, and we're going to find the key that fits that hole. All right? So, for example, AGU, well, we know what letters matches that, UCA. So we're going to look out there in our toolbox of transcription RNA, or transfer RNA letter keys. So here's the tRNA key that has a UCA, all right? That's the only key that will fit the AGU. We're going to slot that key in there. On top of that key is a little green ball. That's an amino acid, and it's a different amino acid for every key. So the GCA key, that fits CGU. The GCA key has a red ball, okay, a different amino acid. All right? Each piece of tRNA is connected, each piece of tRNA is connected to a different amino acid, and we're going to put them in one by one by one by one by one all the way along the chain, and this is actually going to build every 3 letters will correspond, therefore, to a different amino acid. When you create a chain of amino acids, that's what you're building up here. A chain of amino acids is a protein. All right, so this is how it's done. You transcribe the messenger RNA, and then you translate it using tRNA into the amino acids. All right. Pretty complicated, but if you look it over a few times, then it's not actually that many steps. Okay, you take your message, you run it through the ribosome. Every 3 letters using these keys will tell you what amino acid to attach. All right. So there we go. But it is messy. All right. So here's a video that you're probably gonna have to watch a couple times because it goes fast. But here's a video that shows how this is actually done using computer simulations of the molecules involved.
This animation demonstrates how the digital information encoded within DNA is used to direct protein synthesis. This is a DNA double helix containing the digital code which directs the cell in all aspects of operation. And here we see a protein complex called an RNA polymerase traveling down the DNA strand. As it moves down the strand, it carefully unwinds the DNA, preparing it for transcription. Inside the polymerase, we see a single-stranded copy of the original instructions being assembled as individual bases are positioned and added to the growing strand. A stop code marks the end of the protein specification, at which point this copy, known as a messenger RNA transcript, exits the polymerase and heads towards a 2-part chemical manufacturing machine called the ribosome. While the messenger RNA moves towards the ribosome, transfer RNA molecules attach to specific amino acids in preparation for assembly. As the messenger RNA transcript passes through the ribosome, the process of translation begins. Using the instructions encoded on the messenger RNA as a template, the transfer RNA molecules align specific sequences of bases to corresponding amino acids, creating a protein chain. As this chain exits the ribosome, it is met by chaperones which prevent premature folding, while escorting the protein to a barrel-shaped machine called a chaperonin. This machine helps fold the protein into the precise shape required to perform its function. Although it is unclear how the chaperonin achieves this, we do know that accurate folding is essential in order for the protein to accomplish its intended function. Once the protein is complete, it is released into the cytoplasm to do its job.
And that's the protein. It's just an object. So the only thing I think I didn't talk to you about, so to rewatch the video a couple times and it'll make more sense. The only thing I didn't mention is this watermelon-y thing, which folds the protein. So, yeah, the
And
folding
it says
part.
they don't know how it folds the protein.
I don't know. So, I mean, each protein is going to be very different, okay? So the folding is an important part. But anyway, yeah, so you get these things, and that's what a protein is. It's just a shape. And that shape allows you to do certain things. Frankly, that's all tools are, it's just shapes. So different shapes will allow you to do different things. So anyway, a piece of my DNA contains information on A, what my face looks like, or B, what sort of proteins to build in my body. And that's it. B, what sort of proteins to build in my body. That's it, right? It's kind of astonishing. It does not say that I have green eyes or whatever. There is nowhere in my DNA does it say that. It just says, here's the proteins to make, right? That's it. It's kind of astonishing. That's all DNA does. But it works, okay? And it works extremely well. As you can see in that video, it's kind of astonishing that it does work, but it works, right? One gene codes for one protein. It's a direct correspondence. That's what a gene is. It's the instructions for a protein, right? So we have something like 22,000 genes in our genome, in our DNA set. Grapes have 30,000, way more. That's why wine is so much more interesting than most people you've ever met, right? So, so they're actually much more. Chickens are actually not that far behind. We're at 22, they're at 17, almost 1,000. A fruit fly at 15,000. E. coli at 5,000 or 4,000, surprisingly, right? But if you think about it, most of the genes that you really need are the genes to say how to operate a cell, right? And then from there, it's just, okay, now put two cells next to each other. The cell should be a little different than that one. But 4,000 genes required to operate one cell. And then just 4 times that to operate a chicken, right? The flu is 11. Viruses are just basically a little bit of instructions on how to make more of the virus. So it's just the proteins in the virus, which is a coating, a few proteins on the surface of that. That's it. So viruses are incredibly small packages of DNA. So anyway, in Jurassic Park, I know they have like Mr. DNA talking about how you find some DNA from a dino and you use that as a blueprint to build a dino. Anyway, so it's not a blueprint at all. In a blueprint, you have a picture of what you're going to build. If you spill some coffee on that, you could end up building a house with a window in the wrong place or something, right? So it's a picture. Your DNA is not a pictorial representation of what you're going to be at all. It is, like I said before, it's a recipe, right? It says, here's how you're going to build this, step by step. If you look at a recipe in a book, it does not show you what the meal should look like, unless there's actually a picture. But it's just a list of words, right? That does not tell you what a cake looks like. It's just a list of words that tells you how to make it. Similarly, DNA is a list of instructions for how to make the tools that will make you, and that's it. All right. Now, they do have, so you noticed in the video it had an on and an off. There are different codes on the DNA for where to start, where to stop, even for when to do things. So there will be times when the proteins in a cell are not going to be made because it's switched off. Right? Then there will be times when it's switched on. There are actually switches on the DNA. Right? So it affects how things work and are built. It even determines when to build things. Different cells will utilize different tools. So if you're in a skin cell, for example, you look at the DNA, it'll have all the tools activated necessary to use to build a skin cell. If you're in a brain cell, you'll be using different tools. But they all cells in your body have the exact same DNA. They've all got the tool list. The only question is, which tools of that list are they using? If you look at my DNA versus yours, you will find some subtle differences in the tools and in when they are supposed to be switched on and off. And the small, even a very subtle difference can result in a very different building once you built it from a single cell, right? A very different person. So if you have a mutation then, you can imagine the impact this could have, all right? So there's various different types of mutations. Here's a few examples. A point mutation, I showed briefly before. A point mutation is when one letter changes, all right? So a point mutation, you just change an A to a C or whatever. A frame shift mutation is when you shift things out of the way. So from here we have an A and a G, and then we've inserted, so this is an insertion, a T. So now instead of being CTGAGT, it's CTGATGT. All right? We have shifted everything out of the way to make room for something new. A deletion frame shift mutation is when we shift everything together by removing something. So we delete the A, and now we get a GG right next to each other. All right? Now all of these can cause immense change, right? So let me give you an example. So let's say I give you, and remember it always tends to come in 3-letter sets for some reason, so my example is with 3-letter sets. The fat cat ate the rat. All right, the fat cat ate the rat. And now let's try a couple mutations here. Which of these is the point, which of these is the insertion, and which of these is the deletion? So first off, let's look for the point mutation. Which of these is the point mutation? The fat ate the rat, the fat car ate the rat, or the fat cat ate the rat? Which of these is the point mutation? A, B, or C? So the original is the fat cat ate the rat. Now it's the fat ate the rat, the fat car ate the rat, or C, the fat cat ate the rat. Which is a lot of fun to say, let me tell you. I'm having fun with this one. So it's B, okay, car. We changed one letter. One letter change, nothing deleted, nothing inserted, that's a point mutation. It's a very different sentence though. The fat car ate the rat? Okay, explain that to me, right? But that's a very different sentence. Okay, fine, which is the insertion? Which is the insertion? The fat ate the rat, the fat car ate the rat, or the fat cat ate the rat? Which is the insertion? And it is a frame shift. We've had to shift everything out of the way. It's obviously C, the fat cat, we've added another cat. And then fine, the deletion then, fairly obvious. We deleted the word cat, the fat ate the rat. There's something missing. Each of these sentences is pretty messed up, right? Not necessarily a good thing to mess up your words like that. So notice, by the way, that in the insertion and the deletion case, I inserted or deleted 3 letters, right? I took a whole word out. What if I just take a single letter out or in? So here's a worse example. Let's insert a frame shift insert of a C. The fat cat tat F-R-A-T. By inserting one letter, I have obliterated that sentence. All right? It's not even readable anymore. The fat cat tat F-R-A-T. So these can be quite damaging. All right? Doesn't matter how small they are at all. So what effect will they have? Effects of mutation. First off, let's say you normally got, the DNA says TTC. The protein that matches TTC is lysine. All right? If you see a TTC in the genome, that should match lysine. All right? That's the amino acid, sorry, not the protein, the amino acid lysine. A point mutations, okay, so here's all these are point mutations. What would be the different effects that you could get? All right? First off, a silent mutation, if you turn the C into a T, turns out TTT also codes for lysine. All right, so you've had a mutation. It's a point mutation. You've changed one letter. It doesn't even matter because you still get lysine. So that's a silent mutation. Missense mutation would be if you change a single letter. Instead of TTC, it's TCC. and you get the wrong amino acid, arginine, I think, right? What impact will that have? I don't know, but you've got a different building block now. Your protein is probably going to end up being pretty different, right? Which protein is it? Is it a protein that doesn't really matter, or is it the protein that codes for hemoglobin? Because you kind of need that, right? A nonsense mutation will actually turn, for example, TTC into ATC, and that's a stop code. That kills the whole protein, right? You just stop making it.
Hopefully it wasn't something important.
Hopefully it wasn't something important, right? So, what is this likeliest to cause? Okay, so a mutation, what is it likeliest to cause? A, one patch of discolored skin on your body, so you've had a mutation, so now this patch of skin on my body is red instead of normal. B, ability to teleport, could be. Or C, a problem with the functioning of your cells. Okay, so no debate here, it's C, a problem with the functioning of your cells. What kind of problem? I don't know. Depends what you just screwed up, right? But if it's a silent mutation, maybe nothing, which is interesting. So could it cause nothing? Just said it, but yes. Could it be fatal?
Yeah.
Yeah, because you kind of need something like blood. Right?
There have absolutely been fatal mutations. We just don't really.
Yeah, and very often the fetus won't even be able to grow, right? For example, if you don't have DNA polymerase or whatever, you can't even copy DNA. That fertilized egg will not be able to replicate.
I mean, probably a miscarriage or two in there.
So immediate miscarriage. Right. So coming to the X-Men then finally. Here we've got the X-Men. Cyclops has optic blasts. Beast has ape-like strength, size, and flexibility. Angel has wings. Iceman can turn himself into ice, so let's not even touch that one. And Jean Grey has powerful telepathy. Okay, so of those four here, which is closest to already existing human genetics? So I think most of us can agree that it's Beast, right? So he's basically a dude who's extra strong. If anything, he has regressed into sort of like ape-like traits, right? So Beast is most likely. Which would require the fewest gene changes? Still Beast, right? You could have a mutation that results in basically gigantic feet, right? That's okay. That's just growing your feet a little bit different, right? Okay, sure. Growing wings, on the other hand, requires how many genes? A, one, or B, many, many? Many genes. I don't even know how many different tools are needed to build feathers and wings and the whole structure, right? So the muscles to do it, yeah, and everything.
And they have to be wings that can move a human body.
Yeah.
No
Let's not worry about it too much, okay? So basically, one point mutation is not going to give you this, right? The X gene, assuming that a gene could not give you this instead of this, right? No way. A chance mutation granting fully functional optic blasts is A, likely, or B, unlikely? I know this is the sort of question I said I'd never ask, but
Although fully functional.
a fully functional optic blast, yeah, they're not misfunctioning at all. They work, man. So no, there's no chance in hell a single accidental random mutation is going to give you something like that, right? It's fun. I love the comments, but no, not really. So anyway, assuming though that somehow the X gene was able to do all these things, which there's no way, but assuming somehow various differences in the X gene can do all these things, there's no way. But what causes the mutations in the first place, right? Because mutations are real, there are real mutations. So what causes mutations is a real question. So most are natural copy or repair errors. So when you're copying the DNA to make a new cell, for example, or if there's an injury and you need to make new cells, or if you're creating a new sperm cell or egg cell, or whatever, or you're just replacing a cell, because cells age and die, you replace it. So DNA is being copied all the time. And if there's a natural mistake, then that can be a mutation. But I already said that's fairly, well actually fine. The question is that common or be uncommon? So I did say before. So generally speaking, it is uncommon, okay, because DNA copying is really, really accurate. All right, really accurate. However, there's a lot of DNA, so even if it's rare, it does happen, right? Similar to a question I asked before, but this time I'm asking what's least likely? What's least likely? A harmful mutation, B, a neutral mutation, or C, a beneficial mutation? So a beneficial mutation is fairly unlikely. All right, everyone's going C. It's fairly unlikely you're going to get optic blasts, right? But I don't know. You can hope. It has happened in history, right? That an animal had a mutation and it was actually beneficial. This has happened. It happens all the time. So give it billions of years, and you will see examples of it, right? It does happen, but it's extremely rare. Is it necessary for evolution to have a mutation? No. Okay, we already said that. No. Good. Okay, so what are some external causes of mutation? Well, we've already seen one of them. Daredevil was caused by a chemical spilled across him. Similarly, the Teenage Mutant Ninja Turtles, right? Mutagen. The Teenage Mutant Ninja Turtles were actually the Teenage Mutant Ninja Turtles. I don't know if you know this, are a ripoff of Daredevil. Right? Everyone know that? They are a straight up ripoff of Daredevil, which was intended at the time. It was kind of a jokey thing. Their master is Splinter. Daredevil's master is Stick. Get it? Get it? Daredevil fights the Hand. They fight the Foot Clan. Get it? Get it? So yeah. Anyway,
The comic is such a joke.
it was originally tongue in cheek. Anyway, so fine. The Teenage Mutant Ninja Turtles are just Daredevil again, exposed to a chemical that can do it. But there are real mutagens, okay, that we worry about. There are real things. Radiation. Radiation, obviously, we've already talked about this.
Breaks DNA.
Radiation, damage from DNA is likeliest from what? A, radio, B, UV, or C, X-ray? Which of those is likeliest to damage? Because we already specifically talked about radiation's impacts a few classes ago. X-ray, the highest energy radiation, gamma ray, for example. Oh yeah, that'll do it, right? That'll knock out your DNA, but good. So, the X-Men are often called the children of the atom. Remember that they were written in the 60s? So this was a time when worries about the nuclear weapons and nuclear technology was at high. And so it was, the idea of the X-Men is that all these nukes being set off caused radiation, I guess, that led to these mutations. And so now we have the X-Men as a result. So that's the idea behind their nickname, the children of the atom.
As opposed to puddles of.
As opposed to the children of the puddles of mutagens. Yeah, the children of the puddles of the mutagens. Anyway, so children of the atom. So anyway, yeah, now the trick is the cell can repair DNA. As we'll talk about more next class, it doesn't always get it right. So if you force a repair, then you may get an error. So when an X-ray, for example, breaks DNA in half, it will probably be repaired, but it may not come out right. Probably will, but it may not come out right. So that's where you get errors. Right? Now, here's one last thing to look at, all right? How evolution proceeds. In X-Men, it actually starts off, X-Men 1 starts off with this great opening, which is what I want to talk about here. So this video has a lot more to it, but we're just going to watch a little bit of the opening of X-Men 1.
Mutation. It is the key to our evolution. It has enabled us to evolve from a single-celled organism into the dominant species on the planet. This process is slow, normally taking thousands and thousands of years. But every few hundred millennia, evolution leaps forward.
Into some CGI stuff from a company that doesn't exist anymore. Anyway, so okay, so evolution leaps forward. That's an interesting comment, and it justifies the X-Men really well to say that, oh, all of a sudden you can have this something happen, right? So what is Sir Patrick Stewart talking about there, right? When evolution leaps forward, that there is a model for that. It's called punctuated equilibrium, all right? Generally speaking, evolution is thought to be gradual. Makes more sense, like the video with the rabbits, you had slow change, right? Gradualism, slow change. If you've got a butterfly that ends up in two different places, the two different types of butterflies may diverge slowly and steadily until they both look very different 1,000 years later, right? Punctuated equilibrium is the idea that you've got equilibrium, stasis, nothing is changing, and then all of a sudden something happens and you get rapid change, and then you go back to stasis and equilibrium, and then something happens, rapid change, all right? So that's punctuated here's another graph of this. is gradualism is slow and steady, punctuated equilibrium is boom, nothing, boom, nothing, and so forth. Okay? Yeah, Chris?
So would the aquatic ape theory be more punctuated equilibrium?
So I can't remember exactly. That one's not really well supported anyway, to be honest. I know the book talks a lot about it, but it's not. There's a reason why we're not talking about it here. It's semi-popular with some, but it's not really well supported, to be honest. Eric?
So is this like potentially by dormant genes suddenly
No,
becoming active?
no, it's actually really simple. So a lot of people misconstrue this. So X-Men sort of plays on this, to be honest, to say that, oh, you can just have evolution suddenly happen. That's not really what it's talking about. What it's talking about is, first off, it's still over many generations. There's only so fast this can go, right? But it's caused by real reasons. So, for example, an earthquake has just split the colony in half. Okay, now you've got two different regions. One is rocky, one is flat. You know, or some birds have just arrived on an island. Okay, you're going to see some rapid evolution, right? Or an ice age has just started. Okay, you're going to see some rapid evolution. So that's all it's talking about, is that you've got rapid evolution, and then basically nothing happens once you fit the environment well enough, and then something else happens and you get rapid evolution. So fundamentally, this is probably true, but I think a lot of people misconstrue it as saying, you know, basically magic happens and things go wild for no reason, and that's not the case. So in the X-Men stories, I guess the inciting event is the nuclear bombs, right? But in that scenario, if that were to happen, what would have to be the case is a whole lot of nuclear bombs irradiate the whole damn world, and so you get rapid evolution and rapid mutation, right? That's not really what happened though, right? The rest of us are fine, clearly, so.
And everyone would still be dead from the
And
multiple
everyone would be
bombs.
dead, so that didn't really happen. So anyway, what causes the drastic mutations of the X-Men? And of course, there isn't a great answer I can give to that. So like, again, obviously it's fiction, I know, but in universe, is there a reason why? Not really, it's too complex to get optic blasts out of nowhere. And meanwhile, from the same gene, someone else gets, you know, telepathy that doesn't even exist. Like, so it's very, and what would cause it? Because nuclear weapons aren't really that impactful to the whole world. So I don't know, something would have had to happen to the whole world. There are certain things that can affect the whole world, like a gamma ray burst out in space, showers the world with gamma rays, you know? Maybe that's what happened to the Marvel Universe. So here's the last question then, are we evolving? All right, first off, what do you think? Are we evolving?
Can't turn it off.
You can't turn it off, but you can have stasis, right? So it is an interesting question. So here's some examples. Will humanity evolve into superhumanity? Are we going to become superheroes?
Unlikely.
Well, no. So, for example, genetic studies have found genes that have spread over the past 100 generations. So you might think that we've entered stasis and that we haven't changed at all over the last thousands of years, but we actually have in a few ways. Just over the last 2,000 years, for example, lactose tolerance. Okay, me personally, I'm somewhat lactose intolerant, the rest of you are freaks. So lactose tolerance, the ability to drink a cow's milk, is actually kind of not normal, but whoever had that first, they had such an advantage that spread. And it spread over a fairly short time, right? Fair hair and blue eyes also